for us, as Mike mentioned. Oops, what happened there? Okay, back. So as he mentioned, my interest has been on endothelial morphogenesis. And we've approached this question over the years using in vitro systems. So my lab has most exclusively worked on in vitro models using human endothelial cells. And I'm going to try to summarize some of this work very quickly to get at why we became interested in the KRAS story and malformations. So we've developed these in vitro models and using serum-free defined media and either collagen 3D or 3D fibrin gels. The talk today will focus really on collagen matrices. We take QVEX predominantly, add 5 growth factors together. A lot of work went into this. These stem cell factor, IL-3, SDF1 alpha, FGF2, and insulin. Remarkably, these 5 together allow for human ECs to form tubes and networks shown here are vasculogenic type mimic or sprouting assays or aggregates where you can let them sprout out from there. Or most relevant here, perhaps here, is the co-culture where you put endothelial cells and pericytes together and you can form nice, beautiful capillary networks. One of the surprises, was that VEGF and FGF together, for example, or VEGF alone never worked in our system under these serum-free defined media. But if we add these 5 growth factors, all of these things work. The other really nice thing about our system is that you can do real-time movies. These are endothelial cells by themselves, and you can see how they assemble in the presence of these 5 growth factors through matrix interactions. We know this is an integrin-dependent event. The lumen process, the tube sprouting process, all shown as they form this nice network of tubes. So we've been focused for a long time on trying to understand this biology, and you can do time courses shown here. This individual cells assemble into these networks. If you add pericytes with them, notice the difference between EC only cultures by themselves. They're actually wider and get shorter and wider with time, whereas with pericytes, they're much more extended and much thinner. And the reason for this in this result is because when the 2 cells interact together, they actually form a vascular basement membrane. They form the capillary basement membrane. If you don't mix the 2 together, endothelial cells in our hands alone do not make a basement membrane. Remarkably, as endothelial cells form tubes, they carve out a space within the matrix, which we call vascular guidance tunnel. It's an MT1-MMP dependent event. And interestingly, the pericytes get recruited within these spaces, and together they assemble the basement membrane within this almost like a 2-dimensional space in 3D. So this is what the lumen cascade does. It actually creates these spaces, allowing then the mural cells to come in. So as Mike mentioned in the introduction, I've spent a lot of time working on small GTPases. And this is just a summary of a lot of work over many, many years, looking at the key role of small GTPases such as CDC42 and RAC, and now KRAS, and how they interact with downstream effectors to drive this cascade. One of the key things that they do when they form tubes is they polarize their cytoskeleton. They start to target membranes, apical membrane trafficking, which tracks along microtubule tracks, which are often modified by acetylation. They become polarized. At the same time, they've created these tunnels to create these networks. And then that establishes the groundwork to recruit pericytes into the tunnel space, as I mentioned, and then base membrane assembly occurs. One of the surprises for us was when we screened and identified KRAS as one of the key players here, and KRAS is clearly involved. If you knock it down, we see disruption. This is Peter Norton's work, disrupt lumen formation. And then in particular, if you combine it with something like CDC42 knockdown, you essentially abolish the tubulogenic cascade in our system. We also identified in ROS1 a gap for KRAS as something that blocked that. So these small GTPases play critical roles in these events, along with their downstream effectors. We can also then do the same type of assays with pericytes, mixing in pericytes, as I mentioned, and you can watch real-time movies of this, and you can watch them co-assemble. And these are coordinated together quite beautifully. And together, these 2 cells assemble the base membrane and create these elongated and extended capillary networks. So a lot of what we're really studying here is capillary assembly. And I think this is highly relevant, I think, to the malformation story. So when the two cells come together within these tunnels, they assemble beautifully the basement membrane. If you do EC only and stain for extracellular basement membrane proteins, you really don't see it. Only when they're combined, you see strong basement membrane assembly. We confirmed that by EM analysis. Recently, one of my students spent a lot of effort to look at the growth factor control for Perisite recruitment. This is Scott Kemp. We actually found most people in the literature, you'll say, well, PDGFB is really responsible almost entirely for Perisite recruitment. In our hands, it's probably required for about half. But we think actually that multiple growth factors, including PDGFB, D, as well as an endothelin peptide, endothelin-1, TGF-beta-1, and HBGF, we think there's a combined effect. And if we disrupt all of these, we basically abolish Perisite recruitment. Notice that when they do so, the tubes get super wide. The pericytes are actually not really recruiting well at all because you've disrupted these. And notice the endothelial cells are forming fine, but they're just getting wider and wider, almost as if the pericytes are not present. This actually is kind of analogous to what we think we see in the malformations. So our systems really are platforms, if you will, bioassay platforms, in which you can study all kinds of things from tube formation, sprouting, pericyte recruitment. We're interested in vessel breakdown and now the malformations. These wells that we use, micro wells, are 4.5 millimeters wide, 0.5 millimeter thick, and this is actually a gel that's stained with CD31 and pericytes in green, just to show you the extent of this network that's forming throughout this. So we became interested in the anomalies or the malformations because of some interesting things that we've known for a number of years. We're sort of interested in this balance. I'm also a pathologist, so I'm interested in this issue of the sort of balance between morphogenesis and proliferation. And we thought that in the anomalies, and of course, in things like hemangioma, you might have a higher rate of proliferation. We wondered if there might be sort of an inverse relationship between proliferation and morphogenesis. And that's an interesting question that we could discuss more. But one thing we know in our system that I just described is that endothelial cells markedly downregulate cell cycle genes and proliferation during tubulogenesis and assembly. So we were sort of wondering, if you start to disrupt these things, what happens? So what I'm interested in, and what I've been very interested in, is can we model malformations? Can we do so in vitro? And that has been done by Elisa Boscolo with regard to venous malformations, which is very interesting work. And we've been interested in these other malformations, such as arteriovenous malformations or cavernous malformations, or perhaps capillary hemangiomas. Like, how could you model these? And the interesting things for us are, why would the tubes be excessively, say, wide or and not small, narrow capillaries? Why would something like a cavernous malformation actually be a cystic lesion with these large caverns with no extensions or sprouting type behaviors? Or in this case, where you see a lot of cellularity, endothelial cellularity, but not a lot of morphogenesis going on, like you see in capillary hemangioma? Is that because, say, of excessive proliferation? And does that occur at the expense of morphogenesis? So these are the kind of questions that we're interested in. And we were struck by this fascinating work by Jason Fish and colleagues and others, such as the study, and that was the study in New England Journal of Medicine, and then another study, Alalabe in JCI, finding that activating mutations in KRAS were detected in brain arteriovenous malformations. And here, of course, as was discussed nicely by Jason and Strider Meadows, these malformations tend to occur, we think, as a result in capillary issues, and then they become these, tortuous and large vessels, and then allowing communication between artery and vein. So we're interested in this kind of question more from the standpoint of morphogenesis. So what we sought out to do was to express the activating mutation KRAS V12 in the HUVEC cells, and then just did our basic assays, which I've just rapidly gone through, and did normal morphogenic assays in our system. So EC controls, they assemble over time and form nice tube networks. In contrast, when you put KRAS V12 expressing, so all the other point is all of the cells are expressing this. They rapidly form tubes faster, and you can see right away that there's some larger lumen formation even at 24 hours, and they get progressively wider and wider until you get this very widened network. There are no pericytes there, which would normally make this even thinner. But in this case, they get wider. So they seem to be going much faster and wider. And it almost resembles in a way when pericytes are not there, except that it's much more than that. So if you then watch these form over time, this is a stain for collagen 1. So this revealing these tunnel spaces that are created by the endothelial cells when they form networks. You can see that we can see the borders of these in the arrows, and you can see they're quite narrow and the endothelial cells are inside. In contrast, the KRAS line markedly enhanced, markedly faster, wider, and in fact, some of these almost like little circles, and then they become progressively wider until you form these very wide and dramatically widen. And so we believe that this is tunnel formation is predominantly driven by MT1-MMP. So we would predict that these cells have an excessive proteolytic pathway that's driving this. So they excessively widen tubes and tunnels with these cells. So if we quantitate this over time, you can see a marked induction of the lumen mechanism from lumen area, and then real-time movie analysis showing just how fast the KRAS V12 expressing ECs are going. So they're going much faster than controls. We also performed real-time movies, which I think are really useful here. And you can see on the right, the KRAS V12 ECs, which are just dramatically going faster. If you looked early in the movie, you can see that they were actually vacuolating one of the membrane trafficking steps that we have identified over the years. But just look how widened these tube structures are relative to the control ECs. This is actually over a 72-hour period. And a second movie just showing the same thing. You can see all these little vesicles that were throughout, and they fuse together and widen very rapidly. And so you can watch what that looks like there. So a dramatic enhancement. So we think that this is a excessive lumen formation mechanism. We then looked more carefully at the vesicle trafficking type steps early on at different time points. This is by the work of a beautiful work by my student, Juying Sun, who was a tremendous graduate student in the lab. One of the things that we identified, Peter Norden recently identified in the lab, is that caveolin 1 is a strong marker of vacuoles. And you can see that these are, you can see even early on in controls, decoration of some of these structures that mark the vacuole, but also the developing apical membrane. And you can see that these are excessively, more apparent and more driven in the KRAS system. And you can see as this accumulates, and you can see these large vacuoles and then eventually moving into the apical membrane. We also co-stained this with acetylated tubulin antibodies, which we know are elevated in the KRAS expressing cells, and which we think play a role in vesicle trafficking and enhancement of vesicles to traffic to the apical surface. So this is a way for the endothelial cells to rapidly become polarized. So it looks like the KRAS system is going faster, higher power of some of these from this recent paper of Juying, showing these dramatically labeled vacuoles, and then the accumulation of caveolin in this apical compartment as they start forming lumens, shown like beautifully here. And so this is dramatic lumen formation, accelerated lumen formation in the KRAS expressing cells relative to control. Interestingly, one of the expenses of going faster and more rapidly than in the lumen mechanism is actually, we show that it actually strongly reduces the ability of the endothelial cells to sprout. So we do sprouting assays, as I mentioned, in this case from a surface, and you can see that the endothelial cells normally sprout in. And if you go over time, you'll see nice sprouts, but also beautiful tubes. In contrast to KRAS, much more reduced. And if you start looking at these as they actually push into the gel, you'll start to see lumens really quickly. And things like tip cells, which are in the front of sprouts, you really almost don't even see them. You'll end up seeing lumens actually, if anything, pushing in. So that's the KRAS. This is actually looking at the monolayer, and this is a cross-section view. So sprouting behavior is actually in our hands reduced strongly by the expression of KRAS, active KRAS. So we also spent a lot of effort, Juying did, on signaling. So we know a lot about lumen signaling. As I mentioned, there's a lot of key kinase cascades, small GTPases. An important driver of lumen formation over the years is the Sark family kinases. And upstream of that, actually, in our stories is PKC epsilon, protein kinase C epsilon. And interesting, if you look at phospho-Sark over time, KRAS versus control, you can see it's markedly accelerated. We know that Sark is upstream of PAC activation, one of the key effectors of CDC42 and RAC. These are upstream of RAF activation, and obviously KRAS can directly link and activate RAF. But so we see accelerated PAC activation, we see accelerated B and CRAF activation, and we see strong activation of ERK. But interestingly, we also see strong activation of AKT. So this implies that there is some crossover, presumably to the PI3K pathway in this system. Another interesting target that we identified in our studies is this CDCP1, which has been linked to KRAS active expression in cancers. It's also been linked in tumor cells and tumor metastasis by the Heinz lab. And so we see an accelerated phospho-CDCP. I mentioned elevated acetylated tubulin in the KRAS, which we think enhances the assembly of vesicles and transport to the apical surface. We see some kind of modest elevations in MT1-MMP, which is this protease that drives the lumen mechanism. But interestingly, MT1-MMP is known, its surface expression, which we have not really examined yet in the system, but it might be accentuated. And one of the ways that might happen is through Sark. Sark is known to enhance the retention of MT1-MMP on the cell surface. One other key point here, which I think is very interesting, I mentioned proliferation. So using phosphohistone H3, which is a marker of cell proliferation, you can show here rapidly when we put the cells in culture, there's still some undergoing cell cycle, but when we put them in 3D, they basically shut it off. And the same thing occurs with KRAS. So the KRAS mutation, although driving dramatically morphogenesis, we don't think it has anything to do with proliferation. We don't think they're dividing at all. The endothelial cells aren't either, and these aren't either. So we think this is a morphogenic effect of KRAS. We also tried to block this. If you block the MT1-MMP protease, you can completely abolish this. If you block with the Sark inhibitor PP2, it just dramatically blocks the ability of these cells. This is the KRAS cell line that blocks, and they basically end up looking like little, extended tip cell-like things. Interestingly, adding MBQ, which is a inhibitor of CDC42 and RAC, you basically get marked blockade, but also you get little circles which is interesting. We're interested in how do you make cysts? And interestingly, the combination of MBQ plus the KRAS line is interestingly giving you what appear to be some cysts. If you block MEK kinase with the U0123, you can markedly block the system of control, like DMH1, which is an inhibitor of ALK receptors that control BMP signaling, have no effect. So we screened a variety of things, including PI3K, PAC inhibitors, RAF inhibitors, and of course the MEK and so on in a block. Another inhibitor we added was an inhibitor to Notch. The combination of Sark and Notch we've shown over the years block lumen formation quite effectively, and it markedly blocks dramatically. Whereas a lot of other things, such as the PKC alpha inhibitor, a Rho kinase inhibitor, an inhibitor of ALK5, don't block. This is actually a PKC inhibitor, but blocks PKC epsilon, and it blocks. So a variety of things will block this. We also tried to let them form and then try to collapse them. And interestingly, the combination of blocking Sark and Notch together does quite nicely collapse. And you can see many of the same elongated structures. The tube structure collapses quite a bit. Similar story with the RAC and CDC42 inhibitor. So these can form and then collapse rapidly within 24 hours. And adding, for example, the BMP receptor antagonist, doesn't do anything. So that's another experiment then. And I think one of the more interesting ones, in addition, is what happens when you add perisites. And so interestingly, this is the normal system. You can see beautiful CD31 stasis work of Scott Kemp, who helped Jung, who's an expert on Perisite recruitment. You have beautiful tube assembly with Perisite recruitment. Very nice, beautiful Perisites. Note, if you do a tunnel stain to look for where the vascular guidance tunnels, they look nice and elongated generally. And within the, and you can look and see that the Perisites that have recruited to the tubes are within the spaces. In contrast, when you use the KRAS line, the Perisites might slightly reduce the overall lumen width. I think they do. They make protease inhibitors such as TIM3 at some level, which can interfere with MT1-MMP, maybe restrict it a little bit. But the Perisites, many of them are actually off the tubes. They aren't recruiting very well. And you can see here the tunnels again, look how different they look as we showed previously. And then many of the Perisites shown in green here are actually outside of the spaces and are not really recruited in there at all. So we quantitated this. One thing that Perisites do in contrast to endothelial cells during this process is they divide. Endothelial cells have mitogens, these things that I said drive recruitment, also drive proliferation. And the KRAS dramatically does not, they do not expand in number as shown here, hardly at all. And they don't they don't really get to tubes, and they certainly don't elongate very well. So there's many of them. There are some on, but many are off. So whereas in controls, the majority of Perisites recruit. So then the other thing I mentioned is that during this process, when the two cells come together, they make a basement membrane. And you can show beautifully in the system that controls have beautiful basement membrane assembly of laminin, collagen 4, fibronectin, perlecan, nitrogen 1, and nitrogen 2. And interestingly, the KRAS line, which has markedly reduced Perisite recruitment, of course, the tubes are widened, excessively widened. Part of the reason for that may be protease, excessive proteolysis, which might contribute to loss of basement membrane components. And in fact, in particular, you see markedly reduced collagen 4, massively induced fibronectin, and massively reduced perlecan. Interestingly, we've previously published that we think fibronectin and collagen 4 assembly may be connected to each other. And so the loss of fibronectin here might contribute to that loss of collagen 4. And then there is also some reductions in the nitrogens, particularly nitrogen 2. Although these three were the most that were dramatically reduced. We also did some experiments where we just looked at secretion in this system of the KRAS in 3D and 2D, but we'll focus on the 3D. We were struck by the fact that fibronectin was markedly reduced in secretion. We saw a reduction in nitrogen 1, a little bit in nitrogen 2, and also a really strong reduction in perlecan, which is obviously the heparan sulfate proteoglycan, which binds fibronectin and binds and coordinates, the connections between collagen 4, fibronectin, and the nitrogens, and also laminin as it connects to nitrogens. So interestingly, other proteins such as MMP1, which is very interesting, and or TIMP1 or PI1 were not affected. So their secretion was actually, in the case of MMP1, was elevated, in contrast to these, which were reduced. So we think that that may also contribute to why the basement membrane is down a little bit. So this is really the summary of this recent work by Jung Sun. We think that this model system is revealing that the expression of KRAS V12 just dramatically accelerates the lumen mechanisms that we've described over the years, including this vacuole mechanism, this pinocytic vacuole mechanism, which is driven by small GTPases like CDC42, RAC, and RAS, and RAP. These vesicles traffic to the apical membrane surface. This is driven in part by having this accelerated amount of acetylated tubulin, we think. And these membrane surface vesicles are decorated by caveolin 1, and that was can be seen in the controls, but is accentuated. We think these vacuoles come together rapidly, but at the same time, a vacuole fusion like that, there's also a marked, we think, MT1-MMP dependent lumen expansion mechanism that's occurring. And these together, these are all driven by these key drivers of the lumen mechanism, including PKC epsilon. We think Sark family kinases, RAF, PAC, ERK, AKT. We think AKT may be contributing here also, perhaps CDCP1, MT1-MMP, and of course the tubulin that I mentioned, the modified tubulin that I mentioned. If you then if pericytes are in the vicinity, so in order to form a normal capillary network, you need to recruit pericytes. They are necessary and required to make a capillary network in order to keep the tubes narrow, long, and thin, and then to induce the basement membrane. And we think this is totally deficient here. So you have an excessive lumen mechanism combined to a reduced pericyte recruitment and basement membrane assembly. Thus, you have basically immature, capillaries that are not forming properly and not mature. We think that that is largely what the problem could be in this lesions, where the capillary networks are deficient, allowing for the connection of artery and vein. So that's what we believe, that this is ultimately a system that reflects a severe capillary deficiency state, and the vessels that are left or that are in there are excessively wide, which may contribute to the widened vessels that are seen in the AVM syndrome. The other point I'll make here at the end is to say that the other advantage of our model is that we can study many different models, like different mutations. If we wanted to introduce different activating mutations, such as PI3K, which Mark Kahn's lab recently identified as a key driver of CCMs. If you put that in there, what would you get? How would the morphogenesis be different? Or if we used other key genes that have been implicated, say in hemangioma or other lesions, could you then study basically do the same thing? And I think you would almost really have to do that to elucidate this system. The other point to be made is that this might then apply to other endothelial such as lymphatics, which we're interested in as well. So in the long run, I think that's why I think this approach is an important and useful one in the field. So the work that I described today was primarily performed by Jung Sun, a remarkable PhD student. In fact, all of the work that I described were by PhD students, and I've been very fortunate at USF. I have these three, in particular, Jung Sun, Scott Kemp, and Prisca Lin, all participated in this work, and we're all extremely thankful for them and all of the people over the years who've helped my lab and develop. And I mentioned the work of Peter Norden, and I didn't really mention Amber's name, but Amber Stratman, a very major contributor to the development of our system, which we're using now routinely. I didn't mention Daejun Kim, but he did some important work actually on the tubulin cytoskeleton and its relevance to lumen formation, and now that's becoming more and more important. And these are some of my collaborators. I have currently some collaborative work on malformations, and my collaborators on this are Mark Kahn and Chris Hughes. And work over the years with Ondine Cleaver, one of Mike's colleagues at Southwestern, who's been a longstanding collaborator. I actually was very fortunate to work with Elisa Boscolo too, who we helped in a minor way with her elegant work on the venous malformations. So I'll stop there and happy to answer any questions.
Great presentation. And there are several questions in the chat already. Before we get started, I'd like to ask a question.
Sure.
I think it's really interesting, this MMP activity you're seeing. And there are a few reports that MMP activity is elevated in patients with AVMs. I'm curious about the histology of AVMs. Do people see, are you aware, are there basement membrane defects in these lesions?
I believe so. And I think there's certainly, for sure, reduced pericyte recruitment. I mean, there's reduced pericyte assembly, and we think that they go together. So yeah, I think the MMP issue is a very important one. The MMP that I focused on here is MT1-MMP, which is a membrane-associated MMP, and is more involved in this really localized matrix degradation, but certainly it would be involved in destruction of basement membrane components. In terms of like overall, say, surrounding matrix loss, usually that's catalyzed by other MMPs that are secreted. And so that's an interesting question in terms of like which MMPs and how important is that? I mean, matrix integrity is a critical part of vessel formation, but also vessel stability. And if you disrupt basement membrane, I mean, basement membrane, but is obvious, but if you disrupt the surrounding matrix to some extent, that would also be an issue. And that could be relevant, actually, in the pathogenesis of these going forward.
Great. Thank you. What I'm going to do is manage the chat. So
yeah, sure.
when I call on people, if you would like to unmute yourself and show your video, and then you can ask George your question. So we'll start off with Matt Warman.
Hello there. There's a couple of us in the room, so we're masked. That was a fantastic talk. So actually, I have two questions. One question is whether you're expressing mutant KRAS at endogenous levels or at supraphysiologic levels. And the reason why I ask is because we've made some mouse models, we and others, where you express mutants from either the ROSA 26 locus or from the endogenous locus. And using the exact same Cree driver, the phenotypes are completely different based on the level of expression. So my first question was, how are you expressing? And then if you don't mind, Mike, I would like to ask one other after that.
Yeah, I mean, we're using a lentivector, and I'm trying to remember the promoter, but it's not a super strong promoter. It's not a CMV promoter, so it's a little bit lower. I mean, comparing it to the endogenous locus, I don't know comparably. It's not dramatically induced, I'll say that. So I guess the other aspect of this is that all of our cells have the KRAS, and in some of the other models, only a subset of cells, perhaps, like Jason Fish, his very nice talk a couple months ago, was saying that only a subset of cells have the mutation anyway. That's something, by the way, we could do. It's an interesting experiment. We haven't done it. Like, what would happen if we put in 25% endos with KRAS and mix them in with normal cells? I think that would be actually a really good experiment. We haven't done that. So I don't think it's that excessive, I'll say that. But we also, the other aspect is all of the cells have it.
Yeah, so Lisa had asked the question about like mixing amount of cells, and I think 25% cells might be too high. I think you might want to go down to 1% to 2% mutant cells, because it's always been a puzzle, right? How does one mutation in a progenitor cell give rise to this one lesion when most of the other cells are wild type? Even some of the cells in the lesion are wild type. So my other question related to the beautiful Western blot data that you showed. And you made the point that it's the pericytes and the endothelial cells that together are really helping to make the basement membrane.
Yes.
So when you're seeing protein expression changes, how do you know that they are endothelial cell in origin as opposed to pericyte in origin?
Well, the blots that I showed there were endothelial cells by themselves. So that's how we knew that they were So we didn't have the pericytes in there on those blots. But that's a great, when you put them together, endothelial cells are predominant. So they're, the pericytes are probably 20 to 25%, maybe get up to about a third of the total cells. But they definitely make all of the basement membrane components. So they make all of so I think they truly work together to do this, although endothelial cells are stoichiometrically more, so they would presumably control more of it. But I think they're both, for example, the one thing I'll say is that I think that one of the key drivers of endothelial-based membrane assembly is fibronectin assembly, which I referred to, and that is, remarkably deficient in these cells. And it's kind of interesting, classically, if you go back to oncogenesis, the classic oncogenesis works, as cells became malignant, they lose the ability to deposit fibronectin on their surface. I mean, if you go back to the original work with fibronectin, it's kind of interesting, KRAS being one of the most important oncogenes, it's kind of interesting that fibronectin is so deficient. That interests me a lot. But fibronectin is very unique in that it is a mechanosensitive protein. So if you pull on it, this drives its assembly. So we actually think that the mechanical forces between the two cells as they assemble together is what's driving this. So we think pericytes, by their mechanical ability coupled to endothelial cells' mechanical ability, is what's pulling on fibronectin to deposit it. We think it then sort of follows a lot of the others. I mean, it's known that base membrane components can largely self-assemble, but they clearly work together too. So we think fibronectin may be really coupled in our belief to collagen 4 also.
Thank you.
Yeah.
Great. So Jason, can you unmute yourself and ask your question?
Yeah, hi. Thanks for a great talk. I'm sorry if I missed this, but for the culturing, how do you do the culturing? Is it on flat or I guess?
Well, so the endothelial cells are grown in like regular plastic and with endothelial growth media, but then we put the cells under serum-free defined conditions in microwells that have 3D collagen gels. So it's actually 2.5 mg per mil collagen 1, and the cells are suspended in it. And we either would suspend the control endothelial cells or the KRAS expressing endothelial cells, and then we mix them in with or without parasites. And under these, and then we add defined recombinant growth factors, which are necessary for us to drive for this to work and us to survive and form tubes. So that's how we do it.
Okay, thanks. Yeah, I was just wondering, have you tried anything that simulates maybe microgravity or?
No, that's interesting. No, we have not. Good question. I don't know.
Thank you.
Great. Jimmy, can you unmute yourself and ask your question?
Yes, thank you, George, very much. I enjoyed the talk. I think you kind of already covered my question with Matt's question. I agree. I'll just say, I think given the modeling of human vascular malformations and the knowledge that the very low fraction of cells possess the mutation, I think I agree with less than 25%. And I think that particularly if you could label them such that you could see the mutant cells perhaps signaling to the non-mutant cells and see if you see some of these same effects, I think that would be very, very interesting.
That's a great, that's something we really need to do. One of the things I'll say right off the bat, so the tunnels that are created by these cells, once those form, the normal cells will follow within there. So I actually think that, it's a great question. So like what percentage of what would be the sort of, in our system, the minimum number of cells that would be necessary to create an abnormality? I think that's something we have not done. And I think you're absolutely right about this. This is something, obviously what we did is is an extreme, but they individually are really powerful cells, these KRAS cells. So maybe they could carve out, if they start carving out these big spaces, the other cells would look just like that. They would get wide if that ends up being the predominant pattern. But the question is, what percentage would you need to add? So I don't know. We need to do that. That's a great important experiment.
Thank you.
You bet.
All right, Phil, if you want to ask your question.
Yeah, hi, George.
Hi, Phil.
Yeah, you implied that the vacuolation that occurs in the cells that express the active RAS is the end result of an endocytic process. Is that correct?
Yes.
And I was curious, therefore, if you knew what or had thought about what kind of endocytic process it is from the perspective of the large amount of literature that's come out over the last decade showing that oncogenic RAS drives macropinocytosis.
Yeah, I mean, I think that's a great question. I mean, we definitely think this is related to macropinocytosis because it's a CDC42 rack. I mean, you'd have to go back to our first paper actually on 3D collagen morphogenesis, 1996. We show that the vacuoles that we observe, we did this overall culture system a little differently, but it was definitely pinocytic. These vacuoles are heavily pinocytic, and so I think it is likely to be very. And we've showed, a few years after that this was heavily driven by CDC42 and RAC. This whole process. And so I think it's most consistent with what you're saying, which is a macropin. So I think vacuole formation, which is a part of lumen formation, is actually related to macropinocytosis. I think that's likely to be at least a key mechanism.
Okay, thanks. Let me sneak in a very quick other question.
Sure.
In relation to the absence of collagen 4 in your cultures, or much reduced amounts of collagen 4. As you know, when RASGAP is disrupted in mice, the salient phenotype is the paucity of collagen 4 in basement membranes, and that's endothelial cell intrinsic. And it relates to misfolding of collagen 4 in the endoplasmic reticulum, and it's trapping at that site, in fact. So your interpretation instead is that the absence of collagen 4, or indeed even fibronectin, relates to the interruption of perisite interaction with the endothelial cells, and that through some undefined mechanism that's unclear at the moment, that perisite endothelial cell interaction is necessary in some way for collagen 4 export. Is that correct?
Well, in our system with the KRAS expressing cells, we felt when we screened for release, we thought collagen 4 was releasing fine. And we didn't do we have never looked at like what you've done with ROS1, so we have not looked at that. So I don't know, but the KRAS active KRAS, it looks like collagen 4 can be secreted by endothelial cells, and obviously pericytes would be able to secrete it also in our co-cultures. But I mean, we think that at least part of it is that pericytes are not recruiting properly, and then I would also say that there's tremendous morphogenic driving still occurring, which I think results in matrix proteolysis locally by things like MT1-MMP. So I think collagen 4 can be degraded by MT1-MMP probably readily in the system. So I think that can strongly account for what we see also.
Mm-hmm.
All right, Joyce, can you unmute yourself and ask your question?
This is Joyce Tang. Thank you very much for that wonderful talk.
Thank you.
I take care of patients, so I have two clinically relevant questions. I was wondering whether you have looked at the basement membrane in some of these clinical samples, whether what you're seeing in the tissue culture reflects what's going on with the basement membrane and the matrix surrounding these pathogenic tissues.
Yeah, we have not done that, but I haven't. Yeah, we haven't done that screen. The only one I mentioned, I think earlier, was that since pericyte recruitment does seem to be reduced in these lesions in vivo, maybe that might mean that that's a likely possibility, but we have not looked at that.
Do you know, maybe another follow-up question, do you know that there are good antibodies or a certain mechanism to look at the actual?
Yeah, I think there are lots of good antibodies against human basement membrane components. So that could be looked at.
All right.
Yeah.
And then the second question is that, other mutations that are frequently associated with the AVM, so like PTEN and RESA, and you already talked to Phil about this, and you think there are also similar mechanism morphogenic changes, or they're via crosstalks, or it's maybe a little different?
No, I think, well, the only comment I can make on RASA is that we published in 2016 just a paper where we were looking at various screens. When we disrupt RASA1 for us in vitro, we see accelerated tube formation. So we do see it's not as strong as the KRAS V12, but it is definitely accelerated. We did not add pericytes at that time. We haven't done that experiment. So with regard to the PI3K type pathways, that's ongoing for us. We have actually, I have a remarkable student, Priska Lynn, who has like seven new lines of endothelial cells with all these different activating mutations, and we're trying to screen their effects. So that's ongoing.
Can I just add in something there about the RASA1? In contrast, and I've discussed this with you before, George, in contrast to what you have described previously with RASA1 elimination in an endothelial cell in vitro, in the animal, when RASA1 is disrupted in endothelial cells, there is no influence on tubulation. We looked at that with Ondine, but contrary to, and there is no evidence for increased lumen formation, and contrary to what you're seeing in these in vitro systems, in vivo, what happens is that all the endothelial cells die. They undergo massive apoptosis, and that can be rescued by various different MAP kinase inhibitors and other small molecule chaperones that promote collagen 4 folding. So there's a definite major difference between what's happening in your system there with RASA1 versus what actually happens in vivo.
Well, I might differ a little bit, Phil, with the comment actually, because I just don't, I'm just going to say our data is very consistent with a lot of different things, not just that, but we have a lot of other evidence that this, if we, even if we express increased wild type KRAS and so on, RASA1 in our hands stimulates tubulogenesis and the endothelial cells do not die. These are human cells. I don't know what's going on. I can't explain what your mouse model is. All I can say is that knockdown of RASA1 gives you more tubulogenesis in vitro in this system. And our systems have been strongly recapitulated for years by a lot of people. So I think we need to just keep a little bit of an open mind in terms of what may be going on. And we also, I don't know, maybe there are some differences between human and mouse, which I believe strongly in, that they may not be the same.
Strider has a question. Do you want to unmute yourself?
Hi, George. Really nice talk.
Hi, Strider.
I was curious about, you talked earlier about these different sort of Perisite recruiting factors in your system. So I wonder if in your experiments, have you looked at any of those with the KRAS and see if any of those are changes, and that's sort of leading to your problems with the Perisite recruitment?
Yeah, it's a great question. So we've looked mostly at the RNA level to date. There's modest decreases. I can just say that, but very modest. So I don't think it can account for it. I don't know the answer. I don't know. That's really an important question. I think there's a chance that they're getting degraded. One possibility is because of this hyperproteolytic thing going on here. And there are other proteases potentially relevant here that could be degrading targets like PDGFB or endothelin or SDF, or not SDF, that's not the right, whatever, something with TGF beta or whatever. It's very possible that there's something like that going on. And that's really an important question. Yes, and we don't really know. That's an important issue, and we just don't know the answer yet.
Thanks.
And we have one last comment. This is from Matt Mormon again, and I'll just read this real quickly. Matt says that he has mice with a conditional gain and loss of function PIK3CA allele, and he's interested if you would be interested in obtaining endothelial cells from those mice. And you can use those as a gain of function allele with flip recombinase, and then you can turn off the mutant PIK3CA with CRE.
Yeah, that's very interesting. One thing I'll comment, I'm always interested in collaborative interactions. One thing I'll say is that right now we don't have a mouse assay system. This is an example. There really isn't other than the aortic ring assay, which is a classic assay developed by Roberto Nicosia. I have never, the mouse ECs that people have obtained, I've tried to work with a variety of people over the years, from Mike Simons, Luisa Rispe, and a lot of people who tried to send us cool cells that they had. We could never get them to work. So although it's really cool, we've never actually figured out how to make them work. So I don't know what that means. I can't explain that. And I don't know whether it's just a factorology problem, like, something about the mouse, not being the same. That's what I mean kind of by saying that they're not They may not be the same exactly. So it's a little hard to say. The human cells work in our hands.
Great. And Joyce just said that mouse ECs are not as robust in vitro compared to human ECs or large animal ECs. Well, I just want to thank you once again, George, for the great presentation. And I'd like to thank all the participants too for this great discussion. And we're looking forward to seeing everybody next month. And hopefully we can begin seeing each other in person at meetings. So the ISFA meeting is happening in May. If you haven't registered already, it's going to be in Canada this year. So you can go to the ISFA website and register. And then the International Vascular Biology meeting later this year in October, I think, in California. So hopefully we can all meet up there. And thank you once again, George.
You bet.
And we'll see you all next month.
Okay.